Okay, hey guys, this is Mr. Kemmis, and you have reached the DNA structure lecture. So all 10th grade biology kids are expected to watch this one as part of the DNA unit. So you're gonna need a couple things to follow along here. So I'm gonna turn on present mode so you can get everything ready that you need. Okay, so if you're on the Canvas page, you should be able to see this here. Let's see. Okay, you should be able to see the DNA schedule here. So yesterday you should have watched this DNA obsession video, and that gives you a good background on the history of figuring out the DNA structure. So make sure you've completed that, checked out the online lab, and answered the worksheet there. And then today, there's quite a bit to do today. So we have this lecture, there's a structured quiz, which I will help guide you through so you can do well on. I'll make sure I cover all that stuff. And then there's an activity and model making. And then the cracking the code is another fun video for you to look at. Good music in that one too. I didn't like it at first, but I kind of like it now. Also at the bottom of this schedule, you'll see the unit vocabulary. I'm going to go through almost all of these today, probably 1 through 13. So there's a lot of vocabulary today, and you're not expected to have these all down, but you should at least be introduced and start using them in your vocabulary, worksheets, drawings, and everything about DNA. So have that in mind. I'm going to come back to that. The other one too, if you click on view notes right here for DNA structure, it says watch lecture. That's where you're in now. If you click on view notes, then you can come back and look at the notes at any time. That's what I'm going to share with you today. Okay, so let's get everything tiled here. There we go. Okay, so this is the DNA structure notes. A warm-up, you could just review a little bit. What is the structure of DNA? What did the scientists discover about it? You don't have anyone to think, pair, share with at home, so maybe just chat out loud, or you could call your dog over, or maybe you could just jot down some notes to yourself. So we had Erwin Chargaff, Rosalind Franklin, and Watson and Crick. So just take a minute, hit pause, go over these guys. Okay, and now we're back. Let's go over the objectives for today. So the first one is just to explain the role of DNA. So why do we care about it? Why is it important? And then explain the structure of DNA. That's the big one, and we expect some drawings there. We expect some labels, and there's some heavy use of vocabulary that goes along with that. So that's number 2. You should definitely spend a lot of time on there. There's a little embedded video here if you want to hit pause with me, and you can go check that out in the notes. It's actually pretty good. It's from PBS or something. All right, slide 5, the DNA basics. So for this one, we want to just give you the background information. Why do we care about DNA? So let's talk about its name first. Deoxyribonucleic acid. Okay, so DNA stands for deoxyribonucleic acid, and that kind of describes the whole molecule. So it's a deoxyribose sugar. There's a phosphoric acid on there, or phosphate group, and then there's also quite a few bases. Okay, the bases that actually contain the genetic code. Unfortunately, the bases don't make it into the name, which always confuses me. Why won't you have the bases in there? That's the main important information part. Where is DNA found? You probably learned about the cell earlier this year, and we learned about the nucleus. Well, inside the nucleus, you're going to find the DNA, and it is the genetic code for all of life. It stores the information for making proteins. It tells the cell what to do, when to replicate, etc. And then it's also called the heredity, or it's the information for heredity, where you can pass on your genes to the next generation. You got half your DNA from mom and half from your dad. Okay, that's what makes you you. That gives you your eye color, your hair color, some of your personality, when you grow, etc. And then let's just talk about the biochemistry of it. Okay, DNA is a polymer. is a long chain of monomers. So we learned about this earlier in the year. We had quite a few monomers there. We had carbs, we had proteins and lipids, and we learned about those. And then we kind of previewed depending on your teacher, how much nucleic acid stuff you got to. With DNA, we're going to focus more on that structure and teach you that all now. All right, so what is the polymer and monomer? The polymer is the whole DNA molecule. A typical DNA molecule could be anywhere from several thousand base pairs long, several thousand nucleotides long, to several million, or maybe even up to 10 million nucleotides long. So they get to be a pretty big molecule. And that all together is called a polymer. At the end of each DNA strain, there is one acid group. So that's where that nucleic acid comes from. And then inside the molecule, you'll have the base pairs, and that's what's going to contain the genetic code. That's the A's, T's, C's, and G's. All right, now the monomer is a little confusing. Kids get a little bit confused about this one. So the monomer actually has 3 parts. So mono means one. Remember, mono means one, but there's 3 parts to the monomer for DNA. Okay, it is a phosphate, a sugar, and a base. So if you have any paper at home, I didn't bring home a dry erase board. This is normally where I would write on the board in front of the class. So instead, I got some markers here for you, and I got some different papers. So if you don't have any of that at home, so go see if you can find that. Or even a notebook and a pencil, maybe some highlighters, that would be useful too. So here's one of the first things I want you to practice at home, drawing this. This is one nucleotide. Okay, one nucleotide. It has a phosphate, a sugar, and a base. And so this is the simple form of a DNA monomer. You could draw more to the chemical structure, and you'll see that in the notes here. But usually we use a circle for the phosphate or phosphoric acid. We use a pentagon. Do you see that 5-sided shape? That's a pentagon for the sugar, or the deoxyribose sugar. It's missing one oxygen group. And then the base either has a double ring structure or a single ring structure. We just simplify that and draw a rectangle. Did you see the 4-sided shape? That's a rectangle. So take a minute, pause it, go practice, put this in your notes. This is one nucleotide, phosphate, sugar, and a base. All right, let's go on to slide 7. Which diagram shows a nucleic acid and which shows a nucleotide? What is the difference? So here we have the different nucleotides. There's 3 different kinds: adenine, cytosine, guanine, thymine, O. And so those are all there for you. You can see the different chemical structures. You don't need to know this detailed chemical structure here. You can see on the bottom of the sugar there, it's missing an oxygen right here. And that's the deoxyribose sugar. You can see the missing oxygen. And then you put those all nucleotides together in a specific order. You can get some genes. You can get lots of genes, and you can get a whole DNA molecule, which we would call a chromosome. All right, here's the nucleotide again. So you can see that's the monomer, the phosphate or phosphoric acid. You can see the sugar, which we call a deoxyribose sugar. And then there's 4 different bases. Okay, we're kind of lucky. There's only 4 bases to the genetic code. So I think how crazy that would be if there's 26, like the letters in the alphabet. That would be absolutely crazy. But we're lucky. There's only 4. And that's in all living things, from the most simple bacteria all the way up to humans. All right, so these bases, let's take a look at these. These are the ones that Chargaff figured out. There's 4 that they're equal pairs of. They're named after what type of nitrogen base they are. And they're guanine, which is G for short. Cytosine is a C, adenine is an A, and thymine is the T. So usually we just want students to know the base capital letter. You probably want to know that thymine and cytosine have that single ring structure, and nitrogen within that carbon ring. You can see the ends on there, that's for nitrogen. And then the double ring structure would be the A's and the G's. All right, and then Erwin Chargaff, he was the American scientist that figured out the complementary base pairing. This is not the complement where we say, oh, you look very nice today, or hi, oh, I really like your science score. You did really good job on that last test. Not this kind of compliment. The compliment we were talking about here is they go together, okay? A always pairs with T, or it complements T, and C always pairs with G. So this is gonna go down the center of the DNA molecule, and those bases are always gonna pair together. This is important because when we're copying DNA, how are they gonna make a copy of it? This is also important when we're passing on genes to the next generation. How would you pass on those genes and still maintain them for yourself? This base pairing rule is really important for DNA to copy itself and to get that genetic information to your children. So how do you remember this? I got a trick for you. Okay, A always pairs with T, and C always pairs with G. I always think of the wireless companies. I mean, you have AT&T, or have you heard of that one? If you remember the AT&T wireless, you can remember A always goes with T. Another way to find this, and some is that one Cricket Wireless. Cricket Wireless is great. If you remember Cricket Wireless is great, you can remember C always complements or pairs with G. So how could we put this on a test? That's what kids are always asking me. What is that going to look like on a test question? Well, we can ask you about the base pairing rules, or we can make you read a piece of DNA and figure out what the complementary base sequence would be. So here would be a typical test question. You want to take a minute and write this down. Okay. If we give you this strand of 9 letters, what would they go with? Okay. Could you use the base pairing rules and pair those up? So let's see, we got AAC, GAT, CAT. Well, I know A goes with T. Let me grab a marker here. You can practice at home too. T, T, G. Look, those are the first 3 letters of the complementary strand. Could you figure those out? Do you see what's happening here? Let's try the next 3 letters. We have GAT. What does that go with? What would that complement with? A goes with C, A goes with T, T goes with A. Did you get that one? Write that down on your notebook. Those would be complementary bases. All right, and the next one is CAT. What does that go with? We're gonna go G, T, A. So the cat becomes GTA, Grand Theft Auto. Should we do another one, or does that make sense to you? Oh, I'm seeing a lot of people. That makes sense. If you need to go back, rewind, watch that again. That's another skill we want you to be able to read base pairs. Okay. So here's another idea. If you're having trouble with this, nucleotides are like the train tracks. They go together, and then the train tracks are going to pair together across. One track is always going to go with the other. They're going to be parallel like that. All right, those go down this. Nucleotides go down the center of the molecule. All right, well, let's put this all together. What about the outside of the molecule? So we're going to, sometimes this outside of the molecule is just called the DNA background. Okay, it has a phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar. That is the backbone of the molecule. And here on the other side, we're going to start at the bottom. Phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar. Okay, so you can see those are going together. They're parallel, but because one is up and the other is down, for the phosphate group, we're going to call that anti-parallel. That's one of your vocab words. So how do you draw that? Make sure you have a phosphate up on one side, missing on this side, and a phosphate down on this side, missing on this side. That's going to show that anti-parallel nature of DNA, that they're parallel, but they're going in opposite directions. So that phosphate sugar is the backbone. Okay, that's going to be the sides. And here you can see it twisted into that 3D structure, which we call the double helix. Okay, that double helix, another vocab word, make sure you know that one. That is the 3D structure, how that ladder is going to twist. It's going to spiral. Some of the metaphors for that would be a twisted ladder, where the bases would be the rungs of the ladder that you would step on, and the phosphate and sugar would be the sides of the ladder. And when you twist that, that would become the full 3D structure for the double helix. Another metaphor for that would also be a spiral staircase. If anybody's seen that in a home or a museum, where you have those steps going up, and you could climb that spiral staircase, that would also be appropriate. Okay, let's look and see if we can draw some of these parts, shall we? Okay, this is an essential skill for you for DNA structure. So get your paper and your markers or your pencil, highlighters, whatever you got at home. Maybe you have a dry erase board. You're better prepared than me. Let's practice. So what if on a test or a quiz, we gave you this prompt? Could you do this? Could you practice a DNA drawing with 6 nucleotides? Take a minute, pause it. Don't cheat and look ahead. Could you draw 6 nucleotides and label the vocab? Take a minute, see if you can practice that. And then we'll come back and go over the answers. All right, well, here's the vocab list again. Let's see if we can do all of this together. Okay, here's what 6 nucleotides would look like. Did you get this at home? You have 1, 2, 3 on this side and 3 on this side. You should not have 6 all the way down. That's a common mistake from students. Or you should not have 6 on both sides. That would actually be 12. That's a common mistake for students. So 3 plus 3 would get you the full 6. Did anybody get that one? All right, pay attention. Where are the bonds? Okay, each one of these lines between the shapes are called chemical bonds. Pay attention to where those are. They come off specific places here on the sugar. So please be careful with that. That's another mistake from students. Where are those bonds? Which vertices or which corners do they attach to on the phosphate sugar? Oh, hey, did you get the anti-parallel part? With the phosphate up here and missing on this side, phosphate down and missing on this side. Did you get that anti-parallel? All right, what's the next vocab word? We had nucleotide. Could you circle a nucleotide? Let's see. You can pick any one on here. And I'm going to use a different color. Now remember, the nucleotide is the monomer, and it has 3 parts: a phosphate, a sugar, and a base. Okay, there's one nucleotide. Take a minute, draw and label that. Okay, how about, okay, I'm gonna pull up this so you can see this a little better. Did you get a nucleotide? Okay. And then I'm gonna keep going down the vocab sheet. Adenine, guanine, cytosine, and thymine. Could we put those down the center? Could we put some letters down there in a complementary way? Use a different color here. These are the bases. So I'm going to put an A, T, and T. Should we do that? That sounds kind of fun. And a T goes with A, and an A goes with T, and a T goes with A. So there you have the bases going down the middle. Adenine and thymine. And if you wanted to keep going, you could add some cytosines and guanines. And that's part of Chargaff's rule, that those bases always pair together. Okay. All right, double helix. How would you draw this as a double helix? I think that's kind of tough. So I always pull that out a little bit on the side. Here's one way to do it. You can do these different spirals. If you draw it like this, you could see the double helix. I'm going to label that double helix. Some students are able to get the phosphates and sugars down these windy paths, and then they squeeze some bases in the middle. If you're able to do that, well, kudos to you. You're a better drawer than me. I usually like to do them separate. Show the phosphate, sugar, and base over here, and then the 3D structure, the twisted ladder double helix on the side. All right, anti-parallel is the next vocab word. Let's see, what do we got here? I think I'm just going to label that on the side here. I'm going to put arrows to show which way the DNA is going. I'm going to label that anti-parallel. So the 2 backbones are parallel to each other. They'll never touch, but they go in opposite directions. So it's called anti-parallel. So add those little arrows on the side there. Did you get those? Okay. And then we haven't talked really about hydrogen bases yet, hydrogen bonding and covalent bonds. So let's label the different chemical bonds here. We have some very strong bonds here. What color have I not used? Purple. Okay. Wherever you see a solid line, those are going to be covalent bonds, and those are strong. Okay, so there they are in purple, the covalent bonds. You can see between the phosphate and sugar, between the sugar and the base, those are covalent bonds, and those are strong bonds. Those are hard to break. Okay. Now, where's their hydrogen bond? Hydrogen bonds are weak. They're not as strong as covalent, so they're easy to break. Let's see, where would those be? Those are going to be down the center of the molecule. And we're going to show those with dashed lines. Okay. Sometimes there's 2 dashed lines, sometimes there's 3 dashed lines. Okay, those are the hydrogen bonds. So let's label those. And that's actually really important for the molecule too. Okay, when DNA goes to copy itself, it can open up it down the middle. It can read the bases and it can add DNA nucleotides in a complementary fashion. So it can copy itself really easy. It just opens up down the middle of it. It breaks these weak hydrogen bonds and for replication. All right, what's the last vocab word here? Number 13, base pairing. Well, we definitely have that. We have T goes with A, and we have A goes with T. So you can see that there. All right, let's switch back to the notes and let's finish those up. You guys are really close to the end. If you need to go back and practice that drawing, I definitely recommend that. That's a good one. All right, I'm going to present a window, which is this one. Here we go. All right, so the last bit here is a couple quiz questions. Nitrogen bases are bonded together with weak hydrogen bonds. Let me see. Yes, those were the hydrogen bonds down the middle. The nitrogenous bases were bonded with those dashed lines. Okay. Here's another summary diagram. You can see that. You can see how the twisted ladder comes together. So take a look at that. It uses the vocab again. All right, and then here's a quiz question for you. Which of the following is not a role of DNA? See if you can figure that one out. Okay. Did you get it? It's E, all of the above. Very good. All right, so check in. Make sure you can do the next steps for DNA today. There's a quiz on Canvas. Can you do the drawing and label it? It might take a couple times to practice. So do that in your notes until you can do that from memory. And if you want someone to check that, you can definitely message your teacher. And then there's some other stuff for you to do here today. Let's go back up to the schedule. There's this quiz on Canvas. It's a 5-point quiz. There's an activity to complete a model. And then there's another short to medium length video here. Make sure you're doing your CCs. 12.1 is a little bit of the history and background on DNA. And then 12.2 is the structure. So that'll take a little bit to do that as well if you haven't done those yet. And then you got most of the stuff done for chapter 12. Way to go. Today is the big heavy day. So work on that one. And if you need more time tomorrow, I think that's definitely appropriate. All right, well, thanks guys. Make sure you like, comment, and subscribe below. And we'll see you online again soon. This is Mr. Kemmis saying bye-bye. Maybe.